So for those of you I haven't met, I'm Jeanne Stachowiak from UT Austin. And today I'm going to tell you a little bit about work my lab is doing, trying to understand how disordered proteins can organize the plasma membrane in various contexts. Um, and I want to just acknowledge my students. You can see my students and postdocs there in the photo, along with my kids, the little ones. But I just acknowledge the lab that everything I will show you, of course, comes from them, and it's really an honor to work with great students. Okay, let's see. What am I? Here we go. Okay, so I'm going to share just two short stories since we don't have a lot of time together. The first one is about how the actin cytoskeleton can bend the membrane. A lot of you will be familiar with the idea that bundled actin can drive membrane protrusion in these little finger-like structures filled with actin bundles called filopodia. But really, the story I'm going to share with you is a little bit about the role of disordered proteins in actin bundling. So it also applies to actin bundles you would find elsewhere in the cell, like in stress fibers, etc. Okay, so the story I'm going to tell you is centered quite a lot on this protein VASP. There are a lot of proteins that are involved in actin bundling. This is certainly not the only one, or maybe even the major one, but there's a reason we got interested in VASP, and that is that VASP has a high degree of intrinsic disorder, and it's also a multimeric protein. So between those two features, intrinsic disorder and multivalency, it has the characteristics of a protein that we might expect to form networks like these liquid-like condensates that we're finding in the literature. So VASP is a actin polymerase unbundling protein. It hangs out at the growing ends of actin filaments, so at the tips of filopodia, where it binds on to filamentous actin with a filamentous actin binding domain, and then uses its G-actin binding domain to add monomeric, or what we call G-actin, to the growing ends of these filaments. And the reason we started working on VASP oh, sorry, before I get to that, you can see VASP here, it's in red. And just to point out, so this is a B161F1 mouse melanoma cell. It's a highly motile cell that's used a lot to study cell motility. And you can see VASP at the tips of the filopodia, those little red fingernails. And you can also see it at the focal adhesions. If you have a clever eye at the end of the movie, you can notice it there. So the reason we're working on VASP really comes down to a clever graduate student, Kristen Graham, who got excited about this protein. Our lab doesn't traditionally work on actin, and it was really Kristen that got us into this area because she was excited about VASP as a protein that might form protein condensates. So when she wanted to work on this, I told her, well, see if you can purify VASP. I thought maybe that will stop her from going off in this weird direction, but she succeeded in purifying VASP, and she did indeed find that it likes to form protein condensates very readily, and that those condensates are interesting. They're extremely liquid-like. They fuse together on a sub-second time scale here, and they recover almost completely after photo bleaching. So they seem to have high surface tension, low viscosity. They're very liquid-like. And then, of course, she wanted to add actin to them. And again, I was a little bit reluctant to go in that direction, but actin is something that's easy to get your hands on. So we went ahead and gave it a try. And that was when I got really excited. So you can see that when you add actin, the actin readily assembles inside the condensates. And something funny about this is that this was supposed to be a negative control experiment. So this is calcium actin, not magnesium actin, which if there's anyone from the actin field, you know calcium actin doesn't assemble very well. But we still saw assembly inside the condensate, showing there's something about the condensate environment that really promotes actin assembly. Another interesting thing you can see in these images and also the little video is that the actin, as it grows, it likes to move to the inner surface of the condensate. And it's easy to understand why that is if you consider that actin has a persistence length of about 10 micrometers. So if the condensate's smaller than that, it doesn't want to take on the curvature. It doesn't want to roll up into a ball to stay in the condensate. So it'll go to the edges where the curvature is the lowest. So if you keep adding actin to a system like this, you get some interesting structures. So at first you just get this peripheral accumulation of actin, which we called shells in our 3D reconstruction. So just a shell of actin inside the condensate. But as you keep adding actin, you see all these filaments. Interestingly, they condense into a ring. And then eventually, if that ring gets more rigid, it will flatten out the condensate into sort of a disc. So at this point, we were really kind of mesmerized by this behavior. We didn't know what it meant. So we contacted our longtime collaborator, Padmini Rangamani. Maybe some of you know her. Maybe she has spoken here with you. I'm not sure. If not, you might enjoy having her. So anytime I see some mechanical phenomenon I don't understand, I talk to Padmini. And Padmini, as is often the case for her, made some interesting predictions. She said, well, what's happening here must be that the rigidity of the filaments is competing with the surface tension of the condensate. And, eventually, if you add enough actin filaments, those filaments won't be willing to curve anymore. They're going to have to straighten out and give you rod-like morphologies. And I have to admit, I was skeptical of that prediction. I told Kristen, these theorists, they make all kinds of crazy predictions. You can't listen to them. But Padmini is an old friend, so we decided to humor her. And so we added more actin to this system. And sure enough, we did start to see some elongated morphologies. And in this movie, you can kind of see what happens here. Kristen was lucky to catch this. So you can see this condensate. It has a little ring of actin and green inside of it. And when the movie starts, you will see this kind of dramatic transformation into a rod. So sure enough, the actin is able to transform these structures from spheres to rods. And Padmini and her team, Danny and Aravind, they made a really nice simulation that kind of helps you see the competition between the surface tension of the condensate and the rigidity of the filaments, where at first you get a ring and then eventually you get a rod. So we had a lot of fun studying these structures. One of the first things we did was just watch them grow, which is fun to do. So on the cover slip, these rod-like structures grow pretty much linearly and at a rate that's about what you would expect for polymerization of actin catalyzed by VASP. If you frap them, you can get an insight into what's going on. So if frap these growing tips, you will see that the VASP recovers after frap. However, the actin only recovers in the new growth. So that's telling you seem to have stably polymerized filaments, and you even seem to have like a polarized bundle of filaments here, which is interesting to think about. You might wonder what happens to VASP all this time. It's going through this dramatic deformation of the condensate into this rod. So to get an insight into that, you can watch these rods as they grow, and you'll see them run into each other and kind of wick together. And that's telling you that there's still a surface tension, which is a liquid-like property, right? So suggesting that VASP is somehow maintaining a liquid-like state through all this deformation. So we wondered, is that important? So we added a lot of PEG crowding agent to the system to make the condensates very rigid so that they no longer recover after photobleaching. And indeed, when we do that, the actin no longer can win the contest. It can no longer make these rod-like morphologies. So it tells us we need to have a liquid-like condensate to support these long rods forming. One of the things that we wondered, going back to that first experiment, is how it is that the condensate environment seems to be so favorable for actin polymerization that we can even get polymerization under conditions where we don't get it in solution. So a graduate student, Caleb, in the lab kind of dug into that, and he looked at VASP. So I told you already, VASP has a G actin binding domain, which means monomeric actin, and then a filamentous or assembled actin binding domain. And his original theory was that what's going on is the condensate is just like a sponge. It's taking in so much G-actin that we get such a high concentration that we are over some threshold and filament assembly will be spontaneous. So to test that, what he did was just look at if he removes the G-actin binding domain, what will happen to the ability to form these long rods, which indicate actin assembly. And what he found is he loses some of that capacity, but we can still get these rod-like structures, which is perplexing. So we thought, well, let's look at the filamentous actin binding domain. And surprisingly, when you when you mutate the filamentous actin binding domain, that's when we really lost the ability to assemble actin and form long rods. And if you think about that, it might make sense. So anybody here who has studied actin probably knows that actin assembly is favorable in certain buffers, but the energetically uphill step, the most demanding thermodynamic step, is to assemble a small nucleus of several actin monomers that will then seed the assembly of long filaments. And what you need to make these small nuclei of 2 or 3 actin monomers is some protein that can bind at the interface of 2 actin monomers. And that's exactly what a filamentous actin binding domain does. So it could be that just the presentation of many filamentous actin binding domains at high concentration is really good for actin filament assembly. Now, if that's true, it ought to be the case that you can just add a filamentous actin binding domain to virtually any condensate and assemble actin. So to test that, we took a protein from endocytosis that other people in the lab study, this protein called EPS15. You can see it doesn't want anything to do with actin in its native state. Then if you add to it at the end of its intrinsically disordered domain, the LIFAC peptide, which is a short peptide that's a filamentous actin binding domain, then you form condensates that behave just like VASP. They can recruit actin, they can be drive actin polymerization, and can be deformed into long rods. So it seems that what's special about the condensate environment for actin assembly is just the multivalent presentation of filamentous actin binding domains. In this system, you can do other fun stuff like add actin branching proteins, since actin, of course, has a large interactome. And we were curious about actin branching because we had mostly seen bundling, and branching and bundling are sort of natural enemies, right? If you have branches, it's hard to form bundles. So we thought it would be fun to generate some competition between branching and bundling. And what we saw was kind of cool and different from what we expected. So I think we just expected we would get shorter bundles, but instead we got these interesting star shapes where we have a hub with a bunch of spokes coming out, and you can see that better in 3D, that there's a central hub with a lot of spokes. And we see more and more of these structures the more ARP23 we add, which is the branched actin nucleator, to the system. An interesting observation is that the ARP is not evenly distributed everywhere. It's mostly in that hub. So what we think we have is a branched actin hub. And then as filaments grow tangentially away from that hub, they can get bundled by VASP. So it's kind of fun to see that even in a liquid-like network, you can have partitioning of different proteins to give you these kind of higher order architectures. But you are likely wondering, what does any of this have to do with cells? And we also are wondering that, is the ability of VASP to form condensates, is that important to cell motility? So a postdoc in our lab, Rahul Sharma, has been looking into that, and he's been using these cells I already introduced, these mouse melanoma cells. So one of the first things he did was just try to understand the role of VASP. So similar to other people in the field, he has seen that it's really important for VASP to be a tetramer. So if you make VASP a monomer by deleting the tetramerization domain, you don't get localization of VASP to the tips of filopodia, and you can't rescue a VASP knockout the way you can with the tetramer, right? You can't restore the number or length of filopodia by overexpressing the monomer. So he used that idea in kind of a clever way. So he replaced the tetramerization domain of VASP with this light-activated oligomerization domain called Cry2, so that he could shine blue light on this protein and see it assemble only when the blue light was present. And this is a fun tool to use in cells. So he takes these mouse melanoma cells, expresses the monomeric VASP, careful to keep the cells in the dark. And you can see the monomer is just in the cytosol. It's not going to the ends of stress fibers. It's not going to the tips of filopodia. And then when you turn on the light, you see it very quickly move to those locations. So hopefully you can see it at the tips of filopodia, and it's easy to see in these stress focal adhesions at the ends of stress fibers. If you look closely at one of the filopodia, you can see it doesn't have a punctum of VASP at its tip, but it quickly acquires one when you turn on the light. So it seems that restoring the tetrameric state of VASP can let it achieve proper localization. But then we also looked at the effect of this light-induced multimerization on the function of VASP in cell motility. So we monitored the number of filopodia, these little finger-like protrusions per cell. There's relatively few of them in this knockout background in the absence of the blue light. But when you turn on the blue light, oh, sorry, wrong direction. Oh, you get a big burst of filopodia within a couple of minutes, this massive increase in the number of filopodia. And you might think that more light would be better, but it turns out that's not true. So if you add more light, you actually get this sort of rigid recruitment of VASP to the leading edge that seems to block the production of more filopodia. So this is kind of interesting that we seem to need a flexible network to promote assembly of the filopodia. So to investigate that a little bit more, we went in vitro. So we made the same protein in vitro. Oh. And we shined blue light on this protein in vitro to assemble condensates. So at low levels of light, you see the condensates assemble when we turn on the blue light in vitro, and they're very liquid-like. They're merging together. Whereas at a strong level of light, condensates emerge, but you see how they don't fuse together? They're kind of stuck together as if they're rigid, right? You can see that here in these time lapses here, that in one case we have rather rigid assemblies, in the other case we have fluid assemblies. So then if you add actin to these assemblies made with different levels of light, remember that we saw rods before with wild type VASP, and that's also what we see if we add actin to these condensates formed under low light, where they're very liquid-like. But if we form condensates under strong light, where they're more solid-like, we aren't able to create these rod-like assemblies. So it seems to suggest that we need a liquid-like state of VASP to promote the assembly of these filopodial-like structures. And similarly, there seems to be an effect on cell motility. So if we make chymographs to look at the leading edge advancement of the cell, basically how fast is the cell treadmilling forward, we can measure that rate in the absence of light. And then when we turn on the blue light, we get a pronounced increase in the rate of forward motility, suggesting that VASP is helping with leading edge advancement when it's assembled in this liquid-like way. But if we make the light too bright, we lose that. So both for cell motility and for forming filopodia, it seems that there's sort of a sweet spot in the assembly of VASP that we need a liquid-like network and not a solid-like network. And if you think about the role of VASP, which is to bring together the tips of actin filaments to coordinate other factors that bind actin and modify its assembly, it makes sense that we need the ability to constantly remodel the leading edge of the cell to have a functional assembly. Okay, so I'm going to briefly share a second story. I think we're doing okay on time, but I'll try to be brief. Another endeavor in our lab is to look at protein condensation on membranes. And a lot of labs have begun, as well as ours, to look at protein condensation on membranes. But one of the challenges in the field has been that it's hard to reconstitute processes that happen on both faces of the membrane at the same time, which of course is everywhere in the cell. So we wanted to find a way to see what happens if you have protein condensates on both surfaces of a membrane. And this is the work of Johann Lee in our lab. And what Johann does is he takes a TEM grid made for looking at protein structure, et cetera, and he doesn't use it for that. Instead, he coats it with a hydrophobic coating, dips it into an oil and lipid mixture, and then dips it into water. And if you do that carefully, you can form a pore spanning lipid bilayer over each one of the holes in the grid. And when you do that, if you add a protein that wants to phase separate, here we have the RGG domain of LAF1, and allow that protein to bind to the surface, you can observe phase separation out in the 2D plane of the membrane. So you can see that happening here. This is now 2D phase separation, not 3D. So we have these disc-like two-dimensional droplets forming right on the surface of the membrane. Now, if you want to have protein attached to both sides, you have to allow the bottom side of the grid, which was right up against the cover slip in that movie, to be accessible to the protein, which you can do just by sitting it on top of a piece of Scotch tape, actually, to prop it up a little bit so the protein can come under the membrane. And if you do that and then zoom into one of those little grid holes, you see something really strange. So if you look at this strange scene, there's a lot going on there. But the first thing I want you to notice is that before, we just had two colors, two levels of green, the condensed phase and the dilute phase. But now you can easily see there are three shades. There's a condensed phase, there's some kind of even brighter condensed phase, and then there's the dilute phase in the background. And if you look closely at this little structure here, you see those condensed phases that are dimmer joining this brighter structure and sort of completing the brighter structure. And if you analyze these intensities, you can convince yourself that what's going on is that we have some kind of coupling or colocalization between condensates on the two sides of the membrane. That double bright phase is about twice as bright as the intermediate phase. So what this movie suggests is that protein condensates on opposite sides of the membrane, they somehow are aware of one another, even though they are not transmembrane proteins in any way. And somehow being aware of one another, they want to be in the same place, which is very strange and not what we expected. And we thought, well, this must be happening somehow through the lipids because that's the only thing connecting these proteins. So we looked at the lipids and what we saw was even weirder. So a lipid dye seems to be excluded from the protein enriched phase. And this is true for many different lipid dyes, head labeled, tail labeled, red, green. So it's not some kind of a Fred artifact or anything like that. But what all these labeled lipids have in common, as a lot of you guys here in this audience will know, is that they're bulky, right? The lipid fluorophore always has a much higher molecular weight than a regular lipid. And we know from all the protein or all the lipid condensation literature that bulky fluorophores often get excluded from ordered lipid phases. So that made us think that maybe what the protein is doing is creating some kind of an ordered lipid phase. And then those ordered lipid phases are restricting lipid movement. And if that's the case, then maybe those ordered lipid phases like to get together to allow the rest of the membrane to fluctuate. So sort of an exclusion effect. This kind of makes sense in light of some work from others in the field. Rumiana Dimova, some of you must know her. Her lab had shown that protein condensates seem to be able to condense the membrane. So that would be one mechanism of locally reducing the entropy, and it could help to drive this kind of coupling. And in line with that way of thinking, we see reduced lipid diffusion in these condensed regions. Recently, our collaborators, Pengyu Rin and our joint student Ketsia Zinga, have been able to observe this coupling in simulations. So they do coarse grain Martini 3MD, and you can see that if they form condensates, these condensates will couple and remain coupled over time. So they're using these simulation trajectories now to try to figure out what are the exact mechanisms of lipid packing or diffusional changes that could be responsible for this coupling. Meanwhile, Johann has soldiered on. He's been trying to figure out how to observe this phenomenon in cells. And to do that, he has built protein condensates on both sides of the plasma membrane and attempted to observe coupling between them. So to build protein condensates on the inner surface of the plasma membrane, he used this tool, BC-Love4, from Lucas Bagage at UPenn, if any of you know him. So what Lucas did is he made this light-activated protein that when you shine blue light on it, can be recruited to the plasma membrane and will form condensates. So you can see it here. It's in the cytosol. You shine blue light and it comes through the plasma membrane and makes all these little condensates. Then on the outer surface of the membrane, what Johann did is he created a little lipid anchor that is GPI anchored that has a nanobody against GFP. And then he adds a ligand that has GFP tagged to an intrinsically disordered domain. So then when you turn on the blue light to form condensates on the inner surface of the membrane, you see what happens to the outer surface. So before he turns on the blue light, the protein on the outer surface is not at a high enough concentration to form condensates. But then when you trigger assembly of condensates on the inner surface, you spontaneously see condensation on the outer surface. So it seems that this coupling can occur also in the very complex environment of the plasma membrane. And our ongoing work is just to understand how this happens, the sensitivity to various types of lipids and anchors and proteins, etc. Okay, so I stop there and take your questions.
Well, very, very interesting talk. Audience, please put your questions in the chat. So I can probably start with a question. So am I understanding correctly, when you show the actin bundles being promoted by the VASP condensates, it's actually the VASP are wetting the surface of the actin bundles that's growing out of that ball condensates?
Yeah, VASP definitely wets those surfaces. We see very clearly the actin is inside the condensate. It's not on the surface, it's inside. But then as it turns into a rod, VASP has such a high affinity for actin, right? So I think it just constantly coats whatever structure the actin deforms into. We've never been able to observe like actin poking out and then getting coated. If that occurs, it's too fast for us to see.
Mm.
People
Cool,
ask me that
cool.
a lot, and I've not been able to precisely determine, kind of the order of operations there.
Uh-huh. So in the chat, we have a question from Miranda Lynch. So she asked, in the VASP actin bundling, the center initiation points for rod formation looks like a bulbous centers. Do you have a sense of the structure of those clumped regions?
Yeah, okay, that's a very good question. Yeah, there's usually some residual condensate there in the middle, and we've been curious about that. So we have some very preliminary work, which I didn't include. I gave this talk at some time ago to a guy named Jim Sellers, who some of you may know. He works on myosin proteins at the NIH. And he said, "Oh, you should put some myosin in there to figure out where the filaments are pointing." And I thought that sounds hard, but if I had a student, Domo, who was willing to take that on. And Domo now has experiments where she can see myosin 5 and myosin 6, which walk in opposite directions. And she can verify that the plus ends are pointing out and the minus ends are pointing in based on which direction the motors go and where they accumulate. And really what we think is going on is that because the plus end grows faster than the minus end, basically, initially, you probably don't have any polarity in that ring structure. And oh, also, we put the myosin motors on the rings and we see they go every which way, clockwise, counterclockwise, so they have no preference. But then when you go through this symmetry breaking event, the plus ends outrun the minus ends and you end up with plus end bundles going in both directions away from that hub. So to answer the question about the hub, I suspect you have a whole mishmash of things there. You have probably some filaments that are still making a ring, maybe some mixed polarity of ends in short filaments, just kind of some gunk there. And then coming away from that, you have these plus end bundles. That's what we think based on where myosin goes.
So I think you have already answered the first question from Ashok. So he was asking, so what happens when you try to add myosin? You already answered that question. And so he has another question. So for membranes, what role does electrostatics play? Is it electrostatic that is just entraining the lipids?
Ah, that's a good question. Okay, so we attach the protein with the nickel and Ta chemistry, though we've used some other chemistries to attach it, and we kind of always see the same result. But we have added a lot of like polytidylserine to the PS head groups to the membrane. And what happens when you do that with RGG as the protein is RGG is a little bit attracted, right, to PS. Um, because of the arginine in there. So when you do that, if you add enough PS, you can eventually force the condensates to dissolve. So there's some competition between protein-protein and protein-lipid interactions. If you make the protein lipid strong enough, you will outcompete the protein-protein, which is kind of, it makes sense. I don't know why I didn't think of that, but when I saw it, it surprised me. And we can also see that in the simulations.
Cool. So I also have a clarification, perhaps clarification type question. So I remember when you show, when the actin rings form in those condensates, uh, it seems to form on maybe a smallish circle. It's like it's in the more ellipsoid shape, and actin seems to locate on the small circle instead of the large circle. Or am I just,
You know, I think it might be an artifact in our reconstruction. We should probably improve it. But anybody who does confocal reconstructions knows you don't have very good certainty about your Z resolution. So if you're not careful, you everything reconstructs as an ellipse. But I think it's
Oh.
really unlikely. If anything, I bet it's the other way because
Yeah,
the
I
condensate
I
will
would
try to
think it's
wet
like a yeah.
the cover slip. Yeah, this is a good point. This is confusing. We kind of just take the default settings from the microscope, but a sphere should be a sphere. So we need to calibrate and shorten our Z projection spacing. And yeah, I think it's an artifact.
Okay, okay. All right. Well,